In each cell of the body there is a nucleus, and within this nucleus there are chromosomes. These chromosomes, which are made of DNA molecules, contain the genetic information for the cell. There are certain sections of the DNA which are known as genes. Genes control specific characteristics of the body, for example, the colour of your eyes. Characteristics are usually controlled by more than one gene, so there are many genes on one chromosome. Chromosomes are arranged in pairs in the nucleus, and in humans there are a total of 23 pairs of chromosomes. In each pair there is always one chromosome from the father and one from the mother. The genes provide the information needed for the formation of new cells. Cells divide to form new cells in a process known as the cell cycle. In the cell cycle there are the following stages. Stage 1 involves preparing the cell for division. The processes that occur include increasing cell mass and replicating cellular structures such as ribosomes. Most importantly in this stage, the DNA is replicated. This means the cell will have double the genetic information it needs. This is important because when the cell divides into two daughter cells, they need to have the same genetic information. The cells need to be genetically identical. Stage 2 involves the nucleus dividing in preparation for the cell dividing. The chromosomes are pulled to either side of the cell before the nucleus divides. This part of the cell cycle is known as mitosis. The final stage is where the cytoplasm divides to form two identical cells. This type of cell division is used for growth and repair of multicellular organisms, as well as in asexual reproduction. This means the offspring from asexual reproduction are genetically identical to the parent. In the early development of humans, all the cells are the same. These cells are known as stem cells and are able to differentiate into the cell types which are required. In the early development of a human, an embryo is formed, and so these cells are known as embryonic stem cells. You can also get adult stem cells, which are found in bone marrow. In animals, these embryonic stem cells differentiate early in development, and they form specialised cells such as red blood cells and muscle cells. When these cells then divide by mitosis, they can only form the same cell type. However, red blood cells and skin cells cannot divide by mitosis at all, and so adult stem cells are needed to replace these cells. Plant cells are able to differentiate throughout life. Meristems are found in the roots and shoots of a plant, and this is where cell differentiation mainly occurs. Specialised plant cells can also redifferentiate sometimes. Stem cells can be used for many different things due to their properties. For instance, scientists are hoping that the stem cells could lead to the cure for paralysis, vision loss and organ failure. This is because the stem cells could potentially be cultured in labs and then used to form specific cell types. However, all of these studies are done using embryonic stem cells, which can lead to ethical concerns. These stem cells will come from embryos which were aborted or spared from fertility treatments. This can be seen as unethical, as some people may treat the embryo as a human and believe that they have not been able to consent to the experiments. There are also the unknown risks associated with the use of stem cells. They could lead to cancer due to their rapid growth and other diseases. Embryonic stem cells are preferred to adult stem cells because of the limited range of cells produced by adult stem cells. Also, with adult stem cells there is a risk of infection being transferred to patients, and the fact that the patient may have to take immunosuppressants to avoid rejection. Despite this, there has been limited success with adult stem cells, including treating heart disease. Therapeutic cloning is where a cell from an adult is used to generate an embryo of the patient. This then avoids the ethical concerns and means the stem cells won't be rejected by the patient's body. The stem cells could then be used to generate new tissues for the patient. The stem cells from plants can be used to form clones of the plants. This is important in agriculture, where crops need to be produced quickly and efficiently.